Okay, so good evening, guys. In this session, we simply want to revise some questions on nucleotides and nucleic acids. Okay, so I hope you've been also doing your own research. Yeah, so these are questions that we are going to do. I got them from your tutorial sheet, from your tutorial manual. Okay, so I'll go straight into the questions. As usual, if you don't understand anything, please feel free to ask. Okay, so the first question there is on the structures, okay, of purines and pyrimidines. Okay, so we know that purines are two-ring structures, okay, that are composed of one, a six-membered ring, and then the other one is a five-membered ring. Okay, and then when we come to the pyrimidines, okay, we know that it's just a single ring and it is a six-membered ring. Okay, so these guys have general structures, and then on those general structures, if you add some substituents, okay, or some functional groups, they will end up forming different purines or different what? Pyrimidines. Okay, so for the purines, we know we have two of them. We have adenine, and then we have guanine. And then for the pyrimidines, they're the ones that are three, right? We have cytosine, thiamine, and uracil, okay, which is found in RNA. Okay, so let's try to draw the general structures for these guys here. Okay, so we'll start with the purines, and then we'll go to the pyrimidines. So we said a purine is a two-ringed structure. Okay, there's one ring with six sides, and then the other one with five sides. Okay, so let me draw it. Okay, so the general structure for a purine will look something like this. Okay, so I can have an N there, which is double bonded to a carbon, which is single bonded to another carbon here. And then it comes down like that. And then, of course, here is a carbon, which is bonded to an H, double bonded to a nitrogen. Okay, so these nitrogens have specific places where they should be put. So I will number it later on. Okay, and then from here you go to another nitrogen. Even from here you go to another nitrogen. Okay, and then from here you have a double bond that connects to a CH like that. Okay, and then here you have a single bond. I forgot to put an H here, which is bonded to this guy. Okay, so this here is a general structure of a purine. So now you have to know how to number it. Okay? Because if you know how to number it, then it will be very easy for you to draw it. Okay, so you start numbering from here. So you say this is 1, okay, and then this is 2, 3, 4, 5, and then 6. Okay, then when you reach 6 here, you go to the next ring. Okay, so this one here will be 7, then this one here is 8, and then you have 9. So the important characteristics that you must identify on this general structure of a purine, okay, is where the nitrogens are. Okay, so you have a nitrogen at carbon number 1. You also have a nitrogen. Okay, so you have a nitrogen at carbon number 1 here. Not carbon number, I mean N1 there. Okay, and then okay, what was I saying? Okay, so I was saying we have nitrogen atoms at position number 1, position number 3, okay, position number 7, and position number 9. So this here is the fundamental thing that you must know. You must know the positions of these guys, and then you can come up with it whenever you're asked to draw anything concerning these purines and pyrimidines. Okay, so this is not the only orientation. Okay, you can orient it in another way, but the general structure for a purine will look something like this. Okay, so the important features is to identify where these nitrogens are, what positions they are. Okay, because they will be very important when it comes to the bonding and everything. Okay, so this is the structure for the general structure for a purine. And then for a pyrimidine, it's just one ring. Okay, so you can draw a replica of a benzene ring, and then you start adding things. Okay, so we know that a benzene ring will look something like this. So you have N there. Okay, and then this guy is here. We have a CH here. And then you also have a CH here. Okay, and then these will connect to a nitrogen like that. Okay, so you need to practice it. You can't know this without practicing. Okay, so the important thing here to know is also the positions of these nitrogens. Okay, so you start from here counting. So 1, 2, 3, you have 4 there, 5, and then 6. Okay, so you can see it's a 6-membered ring. And then on position number 1, we have a nitrogen. And then again on position number 3, we also have a what? A nitrogen. So this is the general structure of a pyrimidine. So depending on a substrate group that you add to this general structure, okay, you end up with one of the 3 types of pyrimidines. Okay, so if, for example, let me say okay, let me say on top here, okay, on top here I add okay, not maybe on top here I add um, an amino group. Okay, if I add an amino group here, then at this point I add a ketone. Okay, or yeah, I can add a ketone here. Then I'll make what we call a cytosine. Okay, so here let me draw it. So if here I remove this guy, and then I added an amino group like that. Okay, and then here I add a ketone. You see that this structure, it will be pyrimidine quite all right, okay, because these positions here are maintained. Okay, but because you have added this substrate group here and this substrate group here, okay, it will be called a cytosine. Okay, yeah, so when you change the orientation, you add other groups here. You can also form thiamine, or you can form uracil. Okay, so play around with the structures. You need to know how to form. So in case they tell you to draw something like it has to be in your head. Okay, it has to be in you. Okay, so these are the general structures for those 2 purines and the pyrimidines. Okay. And then number 2 here states that what are the structural components of nucleotides, and how do nucleotides differ from nucleosides? Okay, so one of these is made up of 3 things, and then the other is made up of what? 2. That's a difference. Okay, so when they say a nucleotide, then it will be a complete set. Okay, there will be a phosphate group, there will be a pentose sugar, and then there will also be a nitrogenous base. Okay, that will form a nucleotide. And then once they just remove the phosphate group, it becomes a nucleoside. Okay, so let me draw a simple illustration here. Let me draw a simple illustration. Okay, so let's say, for example, I have this pentose sugar. Okay, it should be 5-membered. Okay, and then I have this. Okay, as my nitrogenous base. Okay, and then this guy here is bonded to a phosphate group. So you need an oxygen there, and then you have a phosphate group here. Okay, so when there are 3 of them, this guy is present, the phosphate group, the sugar is present, and the base here, the nitrogenous base. Okay, If all these three here are present, then we call it as a nucleotide. Okay, and then once you just remove this guy here, you remove him here, you just get these two here, the nitrogenous base and the sugar. Okay, it will be a what? A nucleoside. Okay, nucleosides. Okay. Yeah, so that's the difference. So the nucleotide, it will be a complete set of the nitrogenous base, the sugar, and the phosphate group. And then the nucleoside, you just remove the phosphate group. You only remain with the sugar and the base. So that's the difference between those two. Okay. I don't know if there are any questions you're free to ask. Otherwise, I'll go to question number 3. Okay, so question 3 here, they're saying states the different types of RNA and describe the role of RNA in protein synthesis. Okay, so we can take it as the role that each different type does. Okay. Okay, so the thing state the different types of RNA. Okay, and describe the role of RNA in protein synthesis. Okay, so we know that common ones that we know are mRNA. Okay, there's also tRNA, and then there's rRNA. Okay, and then the less common ones we have snRNA, and then we also have micro RNA, and then we also have small interfering RNA. Okay, but the most important things that you must know are the first three. Okay, you must know mRNA, you must know how to describe it and the function. Okay, you must know tRNA because it's important when you're doing the transcription, and then you must also know RNA. Okay, because of the ribosome and its importance. Okay, in the nucleus. Okay, so let's try to look at the main ones. So let's look at mRNA. Okay, so what's the function of this guy here? Or how can you describe it? Anyone who can describe mRNA? Anyone who can describe it? And its role in protein synthesis. When you hear mRNA, what can you say about it? Okay, so it's simple, guys. So this is just the messenger RNA. Okay, so the messenger RNA, what does it do? What does it do? Okay, or how can I describe it? Okay, this is just a single-stranded RNA. Okay, that carries genetic information from the DNA to the ribosome. So it's like a messenger. It passes on the information. Okay, from the DNA, it passes it what? The ribosome. Okay, so it carries information, or just write in short, from the DNA to the what? To the ribosome. Okay, so this is how we can describe it. And then what is its role in protein synthesis? Okay, in protein synthesis, it acts as a template, okay, for assembling the amino acids into a protein according to the DNA, okay, according to the instructions that will be given by the DNA. So it will be responsible, okay, or it will act as a template for assembling that amino acid depending on the what, the information that has been given by the RNA. Okay, so here you can say assembling amino acid. Okay, assembling amino acids. Okay, so that's just a brief description of what this does. Okay, and then we also have tRNA. Okay, so tRNA, what does it do? Okay, or how can you describe it? So first we describe it, and then we state the role that it has in protein synthesis. Okay, so tRNA, okay, it's just a small RNA molecule that carries specific amino acids, okay, to the ribosome. So now get the difference. For mRNA, we said it carries what? Information, genetic information, okay, from the DNA to the ribosome. But this tRNA, it carries what? Specific amino acid to the what? The ribosome. So it carries specific amino acids, okay, it takes them to the what? To the ribosome. So we can describe tRNA as that, okay, it's transfer RNA. So it's transferring these specific amino acids to the ribosome. And then what is its role in protein synthesis? Okay, so its role is just to match. Okay, so it matches amino acids with mRNA codons. Okay, during that process of protein what assembly at the ribosome. Okay, remember the RNA got the DNA, takes them to the ribosome for assembly, right? Okay, so this tRNA now will come in, okay, to match the amino acids with the mRNA codons, okay, so that they are assembled at the in the ribosomes here, okay. And then another important type of RNA, okay, that we can talk about is the rRNA, okay. So in full name, this is what we call as the ribosomal RNA, okay. So we can describe this ribosomal RNA as the RNA that forms part of the what? Of the ribosome. Okay, it's part of the ribosome. It forms part of the structure of the what? The ribosome. Wonder it's called ribosome. Okay. Then the contribution that it does in protein synthesis, okay, it facilitates the binding. Okay, it facilitates the binding of mRNA and tRNA. Okay, so for them to bind, it's because of the what? The mRNA, so that they can be assembled and the information they can be can come out. Okay, so it will facilitate the binding of that mRNA and the tRNA. Okay, and it also has another special function. Okay, it catalyzes the peptide bond formation. Okay, peptide bond what? Formation. So in short, you're forming protein, right? So it acts like a catalyst. Okay, so those are the three main important ones. Okay, but the other ones you can try to read through. Okay, you can try to read through. The solutions here. Okay, so you can pass through. Okay, all right, and then let's look at question number 4 here. Okay, so if you have any questions, guys, ask. So let's look at question number 4 here. They're saying illustrates the structure of DNA, okay, and states different conformations of DNA. Okay. So we know that we have A DNA, we have B DNA, and when we have Z DNA, right? Okay, but the structure, we just know that it's just a double strand. It's just a double strand with base pairs in between. Okay, so when they say illustrate the structure of DNA here and state the different conformations, okay, so DNA, okay, so we know that DNA it's a double strand, right? Okay. Okay, so it's a double strand with the nitrogenous bases there. They are pairing. Okay, so the guanine pairs with the cytosine, and then the thymine pairs with the adenine, right? Okay, so you can, let me just write a simple illustration. Okay, so alpha helix, so you can have something like that, something like that. No, I haven't drawn it properly. Okay, something like that, and then something will follow it like that. Okay, so it will have a double strand, and then on this side, okay, it will have a base pair. Even on the other side, to have a base pair. This side, the base pair. Even on the other side, the base pair. Just like that. Okay, the whole of it. That's the structure of the DNA. Okay, so now this their base pairings, okay, they happen in a specific order. So if you have a cytosine here, then definitely there you must have a what? A guanine. If I have an adenine there, I'll have a what? A thymine here. And these are bonded together by what? By the hydrogen bond. Okay, by the hydrogen. Okay, they'll be bonded by the hydrogen bond here until down there. Okay, so that's the structure of the DNA. Okay, so there's base pairing in between here. Okay, and then what else they do? Okay, they said we mentioned the different conformations. Okay, so I said we have 3 of them. So the first one is what we call as aDNA. Okay, so what is this aDNA here? So this is just a right-handed double helix. Okay, so it will be on the right side. So right-handed double helix. Okay. Okay, and then so we are saying this one is the right hand double helix. Okay, and then we also have what we call as the B DNA. Okay, we also have the B DNA. Okay, so the B DNA is the most common DNA conformation and is also a right-handed helix. Okay, so this is the most common. Most common is the B DNA, and it's also right-handed. Okay, and then the remaining one is what we call as the Z DNA. So the Z DNA, its conformation is left-handed. Okay, so it's the one that is different from the others. So left-handed double helix. Okay, so if you have a left-handed double helix, then that will be the Z DNA. If you have a right-handed double helix, then it can either be the A DNA or it can be what? The B DNA. But how do you differentiate the B from the A? Okay. So the A is dehydrated. Okay, they describe it as dehydrated. And then the B is the most common one. Okay. It's the most common conformation under normal physiological conditions. Okay. So mostly you'll find BDNA. And then some other times you can find ARNA. Okay. If you're experiencing dehydration. And then the ZDNA is left-handed. It's a left-handed double helix. Okay. Right, so that's number 4. And then question number 5 is about denaturation, okay, which is just the breakdown of this DNA. Okay, so they are saying explain how the DNA is denatured. Okay, so how is the DNA destroyed? Okay, so in general, you can say DNA denaturation. Okay, is also known as DNA melting. It's just the process by which the double-stranded structure of the DNA unwinds. Okay, like those hydrogen bonds that hold the base pairs together. Okay, they're no longer strong to hold it together. Okay, so instead of forming a double-stranded structure, okay, it will unwind. Okay, it will like is our pombos, okay? Yes. So that's denaturing. Okay. So it can no longer be together. Why? Because of the following causes. Okay, it can be heat application. Okay, too much heat when DNA is exposed to high temperature. Okay, usually they say above 80 degrees Celsius there. Okay, the thermal energy will disrupt the hydrogen bonding. Okay, of the base pairs as they write the DNA will what unwind, or it can be due to the chemicals that you take in your body. Okay, chemical agents. Okay, so chemical agents, for example, like formaldehyde, or let me give a simple example. For example, urea. Okay, can also cause denaturation. Okay, if you have an excess of urea in your body, it can also cause denaturation of the what? Of the DNA. And then other chemicals can also be strong acids, okay, or strong bases, okay, can change the pH of the DNA, and then the result it will do what? It will unwind, okay, it will separate. Okay, and then other effects include the influence of the base composition, okay? Okay, yeah, so the influence of the base composition can also cause DNA to denature, okay? So we know that between a cytosine and a guanine, okay, there are 3 hydrogen bonds, right? And then between an adenine and the thymine, there are 2. Okay, so some DNA regions, okay, that are rich in guanine and cytosine pairs, okay, require higher energy or stronger chemical conditions for denaturation, okay, because of the additional hydrogen bonds that they have, okay, making them more stable and richer than the A to T regions. Okay, so if you have unstableness in the DNA strand now, okay, some regions are more stable, some others are not, it will cause the denaturation to happen. Okay. All right, so that's about DNA denaturation. And then number 6, we can talk about the Chagas laws. Okay, I can see a question on Chagas laws here. So they're saying calculate the complete base composition of a double-stranded DNA strand that contains 32% what? Guanine. So we know that according to the Chagas laws, let me write here. Okay, so this man here, Chagas, okay, one of the most important people in the discovery of this DNA, okay? So he made a rule, okay? So if he made a rule that the amount of guanine in the DNA that is double-stranded, okay, is always equal to the amount of what? Cytosine. And then the amount of adenine is always equal to the amount of what? Thymine. So meaning the amount of G is always equal to the amount of what? C. And then the amount of A is always equal to the amount of what? T. So here now we've been given that this G is 32%. Okay. So if G is 32%, then it means that even the C is what? Is 32%. And then how can we calculate the percent for this and this guy? Okay. So we know that percentage are out of 100. Okay. So if this is 32, this is also 32. Okay. So it means we can add this and then subtract from 100. So we are going to say the total percentage when you add A plus this T, okay, it should be 100 minus the percent of guanine and the percent of what? Cytosine. So 32 plus 32 is 64. And then when you subtract here, you're going to get a 36. Okay, so meaning the percent of adenine plus thymine is what? 36%. And then according to Chagas laws, we know that the percent of this adenine and the percent of the thymine here will be equal, right? So we can just divide this 36 by 2. So what would that mean? It will mean that the adenine has 18%. Okay? Even the thymine also has what? 18%. And then even the the guanine there maintains its 32%. And then the cytosine is also what? 32%. So it's as simple as that. Okay? So that's how you do it. Okay? So you will always be given one. And then from one, you can come up with the rest. Okay. So any questions there, guys? Okay. So let's look at the other question. Okay. Someone has a question?
Up with the 8%.
Yes, ask.
Did you put the 18%?
Huh? Hello?
Did you come up with 18%?
Oh, okay. Okay. So I was saying, according to Chagas rules, okay, the amount of guanine, which is this guy here, is always equal to the amount of what? Cytosine. And then the amount of adenine is always equal to the amount of what? Thymine. Okay? So in this question, we've been given that 32% is guanine. Okay? So since guanine is always equal to the cytosine, it means even the cytosine will be what? 32. So if we add 32 plus 32, we are going to get 60. Okay? We're going to get 64%. And then we know that the total percent is always 100, right? So we are going to say 100 minus this 64. Okay, we are going to get 36. So it means this 36 here, the percent when you add the adenine plus the thymine. Okay, so that is what I indicated, A plus T. Okay. Now, by Chagas rules, we also know that the adenine is equal to this guy. So if adenine is equal to this I, it's the same as we put A where there's T here. We're going to have more like 2A, and then this 2A is equals to what? 36. And then we divide both sides by 2. We are going to get 18 there. And then even for the adenine, we also got to get what? 18. So that's how we got the 18 there. Okay, is that okay? Okay. All right. Okay, then let's look at some base pairings. Okay, so here we have a complementary sequence, which is in the 5 prime, 3 prime direction. Okay, so we are saying write the complementary sequence in the standard 5 prime to 3 prime notation. Okay, with this sequence that we've been given. Okay, we've been given this. Okay. All right, so this is DNA. So we know that the C goes with the G, and then the A goes with the what? With the T. We know. Okay. So let's find for this first one here. Let's find the complementary one. Okay. So we have G, A, T, C, and then AA. Okay, so this is what we have. So we want to find the complementary strand for this guy here. Okay, so what are we going to be doing? We're just going to be pairing them. So the G goes with the C, the A with the T with the A, the C with the G, just like that. Okay? So from here, it means this one here will pair with the C, this guy here will pair with the T, this guy here will pair with the A, the C here will pair with the G, the A here will pair with the T, even this A here will pair with the what? The T. So what we have come up here, down here, is complementary strand. So this guy here is the complementary strand, the one I'm highlighting. I don't know if this is here or yes. So this is a complementary strand for the first one. Okay. And then the other one, okay, so they said the complementary is going in the 5 to 3 prime direction. So it means this one here was starting from the 3 prime, okay, to 5 prime. Then this complementary here is moving from the 5 prime to the what? To the 3 prime like that. Okay. So this is the first one. And then let's look at the B, how is it going to be? So the B, I'll start with the 3 prime here. Then I have A. Oh, no. I have T. Okay. I have T there. Starts with T. So T, C, G, A, C. And then we have 5 prime at the end. Okay. So now let's find the complementary sequence. Okay. The complementary sequence, since we have 3' here it will be 5', and then here the T goes with the A, the C with the G with the C, the T with the T, the A with the T, the A with the T, the C with the what? The G. And then here we have 5', which means here we're going to have what? 3'. Okay, so I am sure you've gotten the hint of it, right? So this is what we do. Okay? So just know that in DNA, the C goes with the G, the T goes with the A. Okay? And then when you go to the RNA, there will be a difference somewhere, right? So in the RNA, you have the C going to the G, okay? And then we don't have the T in RNA. We don't have the thiamine. So it means the A will now be paired with the what? The uracil, which is U. Okay, so in the RNA, A will be paired with U. Take note of it. Okay, and then the other one, I'm sure you can eat it. We have something like A, C, G, A, G, T. Okay, so this one here, you can complete it on your own. Okay, and then question, the follow-up question is the same question. They're just saying draw the schematic representation of each of the following answers in question 7 above. Okay, so I sent a schematic representation in the group. I'm sure you've checked it out. Okay, or if you want, you can just draw it like this. No need to show those base pairs and phosphate group. So you can use this diagram here as your schematic representation, or you can use the one that I sent in the group area. Okay, so check out the one I sent you, or you can choose this, whatever you choose. All right. Okay. Okay, and then what's the next question now? Okay, so here they're saying which is more hydrogen bonded? Okay, which one is more hydrogen bonded between the tRNA and the mRNA? Okay, so the one that is more hydrogen bonded is the tRNA. Okay, so when you form that, what do you call it? I've forgotten it. Okay, so when you form the clover leaf, okay, of the tRNA, or they call it the L shape, there's something that is good one. Okay. Okay, so the clover leaf, or what we call as the L shape, is this diagram that you've seen a lot. Comes something like that, and then here comes like that, like a cross, right? Yeah, something like that. Then it comes here, and then it goes here down where the transcription happens. Okay, and then it comes back like that. Okay, so in this hot shape here, the L shape or the clover leaf, okay, here you have the base pairs here. So all these base pairs here, all these base pairs that will be alongside here, okay, will be hydrogen bonded to the next base pair on the other side like that. Okay. So what I've drawn here is what we call as the tRNA. So they will be bonded here with hydrogen bonds like that. Okay, hydrogen bonds like that, hydrogen bonds like that. Okay. So as a result, the mRNA. Okay, the mRNA, the bonds are not so much. Okay, not so much. So the one that is most or more hydrogen bonded is the what? The RNA. Because of its shape here, it has a lot of hydrogen bonds here. Everywhere here it has hydrogen bonds. Okay, and then this one here should be the simplest for you guys. They're saying state the differences between RNA and DNA. Okay, we know that RNA is what? Is single-stranded. Okay, and then we know that the DNA is double. What other difference can we get? We know that in the in the DNA, okay, we have the deoxyribonucleic acid, right? Okay, the one that is deoxygenated. And then in the RNA, we have the ribonucleic what? Acid. Okay, and then in the RNA, we have the ribose sugar, okay? And then in the DNA, we have the deoxyribose sugar, right? Okay, again, it doesn't have oxygen. No wonder it's called deoxyribose. Okay, and then I already stated the double-stranded, the single-stranded, okay? And then the nitrogenous bases that you find in the DNA, this one here, you find the adenine, the thymine, the cytosine, and the guanine, okay? And then on the other part for the RNA, you find the adenine. Then instead of the thymine in RNA, you find the what? The uracil, okay? And then you also find the cytosine and the guanine, okay? And then we know that in DNA, the A pairs with the T, but in RNA, the A pairs with the what? So those are some of the differences that you can state. And then you can also state in terms of their functions. We know that the DNA stores genetic information, okay? The makeup of an organism. DNA stores the genetic information. And then RNA is involved in protein synthesis, okay? So RNA is involved in protein synthesis, gene expression, and various cellular functions. Okay, but DNA is specifically storing what? Genetic information. Okay, and then you can also mention the conformations or the types. Okay, yeah, you can also mention those ones. And then you can also compare them based on their stability. Okay, so the DNA is more stable due to the deoxyribose sugar, okay, and the double-stranded structure. Yeah. Okay, so it's more strong because it has those double strands, okay, as compared to the RNA that has a single strand. Okay, so it's less stable, okay, because it is single-stranded and it has the ribose sugar. Okay, so all these answers I'm saying, they are there on. Okay, take them out. Then let's look at this last question here. Okay, so here they're saying write the sequence of mRNA molecules. Want to translate this DNA template to the RNA template, right? To the mRNA template. Okay, so what are we going to do? We first have to find the complementary DNA strand here, okay? And then we use that complementary DNA RNA strand to come up with the what? The mRNA strand. So that's what we, okay, so you just don't do direct. Okay? First, we have to come up with the 5' to 3, could be 3' to 5, because here the side is 5. Okay, so we have to first come up with a complementary DNA template for this guy here. So we have to find the 3' to 5' for the DNA, okay? And then we'll use this one here to find the what? The mRNA. So let's, okay, so let me write it on my board. Okay, so that we are with 5', and then we have A, T, C, and G, T, A, C, C, G, T, T, A. And then we have what? 3'. So we must find first the complementary what? DNA strand. Okay, so this would be 3' to, okay, so A goes with the T, T goes with the A, C goes with the G, G goes with the C, T with the A with the T, C with the G, C with the G, then G, is this G? Yeah, so G with the C. Then T, we know it's A, this T again is A, and then A is what? It's T. And then this should be 5'. So this is the complementary DNA. This is still DNA. This is still DNA. And then from here now, we can find our what? Our mRNA. Okay, so let's find the mRNA sequence. So mRNA sequence. Okay, so the mRNA sequence will be from 5' to 3'. It will be the same as this guy here. So we are going to say 5', okay, and then we are going to be using this guy here now to find the complementer. So remember, we said in mRNA we don't have T. So wherever there's A, we are going to be putting what? Okay, we are going to be putting U. So let's do it. So T goes with the A goes with the T, but you don't have a T in mRNA. So we're going to put what? U. Then G goes with the C as usual. This goes with the G. A goes with the U again. T goes with the A. This is GG. Okay, so I have C, C. Then I have C, here I have G. A, I have U, U, and then T, I have what? A. And then I have 3' at the end. So this one here will be the mRNA what? Sequence. So we first have to find the template for the DNA, and then you use that template for the DNA to find for the what? mRNA. Okay, so any questions, guys? Okay, so I think, yes.
Sorry, I just joined some few minutes ago. So if it says 5', meaning you're starting from the opposite. How did you do it?
Yeah, so
Like
if
you're saying 5' to 5'. So if you're drawing like now, since the one that you've written down here is for DNA, so when writing the opposite 5', you swap them.
yes,
Oh, you follow the same sequence. T with A with Shan.
yes, you follow the same sequence, but just know that in DNA, when you have A, it will pair with the T. And then when you are going to the RNA, when you have A, it will pair with the U. So don't make a mistake.
Okay, thank you.
Yes. Okay, so if there are no questions, I'll end here, guys. I wish you all the best. And I uploaded the solutions to all the tutorial sheets in TMS. I don't know, maybe I just missed one for lipins. I think so, but the rest are there, so you can check them through. Okay, so have a blessed night.